All right. Should have just selected the slide. I don't know why I did this the lazy, slow way. Okay. All right. So that brings us to these terms that I'm sure you guys have heard before, but you do need to know them for this course. And like these are some terms that I'm going to be actually using pretty regularly. You'll hear me talk a lot about how a molecule is hydrophilic. So you're going to need to keep up with these terms, right? I talk a lot about this. Hydrophilic versus hydrophobic. The terms themselves tell you what's going on with the molecules, and that's good. So that's somewhere to start with. Hydrophilic versus hydrophobic. I actually went to school. My graduate school was in Philadelphia. It's the city of brotherly love. Makes it easy to remember that philic love, right? So water loving, and everybody knows phobia is fear, so water fearing. Easy. So if water, and we already established, and we're all well aware of water being a polar molecule, and it likes associating its negatives with positives and its positive with other negatives. So it likes polar things. So things that are hydrophilic or water loving are going to be polar molecules. Things that are hydrophobic or water fearing, they don't have polarity. So when we think of carbon, which a lot of life has carbon as its backbone, right? So organic molecules, the whole point of organic chemistry, carbon backbone. Carbon is very, very stable molecule. If you have a carbon with 4 hydrogens around it, it's not going to hog. It's going to be very fair in how it shares its Barbies, right? It's just happy. Everybody's happy, equal sharing. There's no polarity at all with a happy carbon molecule. However, you get a lot of carbons with a lot of hydrogen. Even if you insert some double bonds in there, the carbons are still happy to associate with one another and double share 2 Barbies between them. All of that, it's so stable, there's no charge at all. There's no favoring, there's no electronegativity. These guys are just all happy, good, shared equal. You get a lot of that going on, you get hydrophobic situations. So these long hydrocarbon tails that we get make up structures of things like fats and waxes that we already know are like water repellent. They don't mix with water. The whole reason they don't mix with water is because they don't have polarity. So that carbon, hydrogen happiness that's going on is great for carbons and hydrogens. Water, that oxygen, that electronegative oxygen, it doesn't want to have anything to do with that, right? So they don't get along. Polar and non-polar molecules do not get along. And that's what defines our hydrophilic and hydrophobic, okay? So if you ever hear me talking about, oh, this molecule is hydrophobic, you should know automatically, must not have very many oxygens in it, probably has a lot of carbons and hydrogens. So you can say that automatically about the chemistry in it, and you know how it's going to interact with water too. Does it in the name, but also you can think about it at the chemical level. Amphipathic is a term for these molecules where we have both going on. So we will have a phosphate group, for example. Now, I'll tell you what about some phosphate. Phosphate is one of these molecules that has, I wish I had, I don't think I have like the pen for this. Oh, I did. Awesome. Okay, great. You get to see some art now. And sorry, I did some more. All right, so phosphate looks like this. So this is phosphorus. It's just P. That's not the same as phosphate. Phosphate looks like this. I'm not good at knowing how many bonds the phosphorus makes. Okay, so there might be more or less double bonds. Don't judge. But it has lots of oxygen. There's 4 oxygens around it, and there's double bonds involved, okay? And then somewhere on this other oxygen, this guy is bound to whatever. So it's probably a lot of carbons that probably have a lot of hydrogens. For example, okay? Now, and this would just continue on, and you guys know how organic chemists like to do this. And at each point you have a carbon, and there's some hydrogens flying off that you just know are there because of drawings, I guess. So whatever, so you guys get it. So this would be an example of where you have, man, anytime there's an oxygen, that oxygen is always going to have electronegativity, and it's always hogging. Just bank on it, okay? So how many oxygens have we got going on here? 4. All of them hogging. So when we think of a phosphate group, this is just a situation where you're going to have a freaking negative charge. So you're going to be electronegative heavy here. These guys actually have some hydrogens that usually associate with them, but they'll ionize off. And so we have a negative actual ion at the end of this. So it's a true negative charge here connected to this hydrophobic tail. And then this one is hydrophilic because it has charge. So if it's straight up charged or if it has a polarity, right? If it's positive or negative to it in any way for whatever reason, then it's hydrophilic. If it doesn't have any charge to it, it's probably hydrophobic. We can even see this in smaller molecules like ethanol. So I don't know if you guys remember anything about organic chemistry. Gotta love it. You have the methyl, ethyl, propyl, butyl. That's my rule of where I start with every time. So methyl is 1 carbon, 2 carbon, 3 carbon, and then 4 carbon. So ethyl alcohol or ethanol means that you have 2 carbons. One of them is connected to an OH group at the end. So what's happening with the rest of it? It doesn't tell you in the name, but it's all just hydrogens. So this is ethanol, pretty basic compound. It's just 2 carbons. You got an OH group. Do you think this OH group has polarity? It has an oxygen. Guys, yes, it does. It has polarity. So that oxygen will be negative no matter what. Even if it's still connected to that hydrogen, that oxygen is still stealing from that hydrogen. Okay, so that oxygen there will have that negative polarity. That gives it the ability to mix well with water. That's why alcohol mixes well with water. But you ever notice how it mixes a little weird with the water? It doesn't like quite always just immediately go into solution, especially if you have a high concentration of alcohol. It sort of beads a little bit, but it mixes fine eventually. That's because of this part. So ethyl alcohol is very unique in the aspect that it has a little bit of hydrophobic aspect and hydrophilic aspect, just enough that it can kind of straddle the line. And so that makes it useful and combining with organic molecules. And we'll talk about how it's effective in getting rid of, bacteria and stuff like that later on. That's like a chapter 11 thing. But we're starting to see the reason for looking at chemical structures now and kind of understanding that organic chemistry that we learned about before this course and why that will be useful for us understanding how these molecules are going to interact with each other. So if you think we're not going to talk about how ethanol affects membranes and things like that, well, we are, right? Because what is this molecule that I've drawn down here? It's not a very good drawing. It's not entirely accurate, right? But what does this represent? You guys know what it's called? A phospholipid, right? You guys know what phospholipids are for? They make up your membrane, your lipid bilayer of your membrane. You guys know how that works? So you'll have your phosphate heads. They're all polar, so they are going to kind of naturally want to just be polar, would rather be near even opposite polar than near hydrophobic. I mean, it's that much of a distinction. So those tails of the, this actually has 2 per head, but we're not going to worry about that right now. The tails will want to hang out with each other so much, and the heads would rather hang out with each other and the water on the outside. So they are sacrificing themselves to hang out with the water so the tails can be contained within on the inside. And that's what makes up your lipid bilayer. That's why it's a bilayer, because you have a layer on top, first layer, and a layer on the bottom, that's your second layer, lipid bilayer. That's what makes a membrane. This is really important to understand that because you can now kind of get the sense of how something like ethyl alcohol could mess that up because it straddles the line just enough. Maybe it can break up those interactions between these guys, and it does.
That's what's around the cell.
Yep, this is what is going to be around every cell. So what defines a cell, right? A cell is a membrane-bound unit that is capable of duplicating itself. It has all the necessary things that it needs to make a copy of itself, but they're all membrane-bound. What's a membrane? That's a membrane, right? We were talking about pH. We already got into this, so I'm not going to go over this again. We talked about that scale and kind of how acidity works and what that chemical equation means to us. Here they can actually write it out better than I want to count like I do, but you can see there that when we talk about 14, we're actually talking about 10 to the negative 14. It's a much smaller number than at the other end, right? So keep that in mind when we talk about pH. Carbon, very happy to make 4 bonds. As long as it's making 4 bonds, it's completely and totally stable. It'll bond with pretty much anything and be happy. The carbon will be happy, and pretty much anything with it will be happy. It's important to remember that in organic molecules, in order to have an organic molecule, it's usually carbon-hydrogen backbone. The carbon is what makes it organic. But if it's not binding with hydrogen in some aspect, it doesn't necessarily qualify as organic. So when we look at calcium carbonate up there, the third one on that top list, not an organic molecule. It has carbon in it, but there's no hydrogen associated with it, so we don't technically call it organic. Carbon dioxide is one of those things that kind of straddles the line too. Now, it's so commonly involved in biology that we consider it to be part of organic systems, but technically, according to the rules of organic chemistry, because there's no hydrogen, it's not really an organic molecule. Okay. We're looking for the hydrogens there too. So that's important. Keep that in mind when you're answering your quiz questions. Cool. I'm not going to make you guys memorize all of the little functional groups, but I will talk about them sometimes. So if I start talking about an amino group and you don't remember what that is, please call me out. Okay. Because if you don't know and you think you're the only one that doesn't remember, guaranteed almost everybody else in the class doesn't remember either. They're just not saying. Right. I don't always have a good grasp on that stuff. So you guys have to call me out if I'm not seeing it. All right, so let's get into our macromolecules. Macromolecules basically just are major groupings of these biological molecules that are important in our biological systems. And each of these macromolecule groups consists of repeating units. And those repeated units are called monomers that come together to make the polymer macromolecule. Okay? So each of these units has some sort of monomer. The only one that's super, super vague is going to be our lipids. Okay, those are really, really vague. But we kind of get a sense of what makes a lipid because they've got those hydrocarbon tails. It's the only one that has the long hydrocarbon tails. Okay, they're the only ones that have distinctly, definitely hydrophobic aspects. The other groups, they don't have that. Okay, so what's our first group? The carbohydrates. Carbohydrates consist of sugars and basically polysugars. So what's a word for sugar? Saccharide, because we have to be fancy when we're in science. So polysaccharide means you have multiple sugar units linked together. So starch, for example, just having glucoses linked, just over and over again. Glucose and there's other sugars in there too, but we all kind of know about glucose, and we're all probably aware of glucose's importance in biology, even though if you're not entirely aware of its chemical importance in making energy for your body, which that is a chapter 10 issue. But we're aware of the oses. Say glucose, sucrose, lactose, fructose, os. Os means sugar, okay? So you hear it? Galactose sounds like galaxy, but it's sugar. So galaxy sugar, and that's fine if you need to think of it that way. But if you hear that os at the end, that's a sugar. Ribose or deoxyribose, even those are sugars, okay? They're 5 carbon sugars that help make up the backbone of DNA or RNA. So those are still important. Whereas things like glucose is a 6 carbon sugar, fructose is a 5 carbon sugar. The only ones that I need you to remember that they are, how many carbons they are, I'll tell you guys, okay? So obviously you should probably know that ribose is a 5 carbon sugar. If you know that ribose is a 5 carbon sugar, so I'm gonna talk about it a lot in this class, then you know deoxyribose is a 5 carbon sugar because it's just ribose, deoxide. So literally we just took off an oxygen. It's in the name. Okay, so you know it's 5 carbon because ribose is 5 carbon. And it doesn't say decarb, does it? It just says deoxy. So it tells you. And then glucose. So ribose and deoxyribose, 5 carbon. And then glucose is 6 carbon. The importance of that 6 carbon will come out when we start talking about metabolism. Okay. And you guys taken physiology yet? Yeah. Okay. Then you probably have an idea of aerobic respiration. It's my favorite topic of all time because I think it's the coolest. And if you guys don't know, or even if you don't think it's the coolest, you'll probably hear about why I think it's coolest. Hopefully I can sway you because I think it's pretty cool. Okay. We'll get to that in chapter 10. But here's some pictures that I'm never going to have you draw, recognize, or anything, but you've probably seen them in chemistry, so maybe it'll be a little familiar to you. Sugars. Okay. Lipids now. Lipids have those hydrophobic aspects to them because we have those hydrocarbons. So if it's hydrogens and carbons, we're very happy, we're very stable, we are not polar. So not polar means hydrophobic. And so that hydrophobic aspect is really what's characteristic for our lipid groups. Anything that you would consider in your mind to be water repellent is probably going to fall into this group. I'm not talking about like actually it's water repellent because it's solid. I'm talking about like if you were to mix it as a liquid with a liquid. So obviously, like things like wax, we know wax is water repellent. We put it on cars specifically because of that, right? We know fats like oils, like olive oil, doesn't mix well with water and all of those. So oils, fats, and all those things that we're already aware of. We talked about the phospholipid. You guys saw the structure for that. Yeah, the lipid part literally is hydrophobic. The phosphate part is the top of it. That gives it that amphipathic. So amphi, just like amphibians can be on the land and in the water, they can do both. Amphi means both. Every time you hear it in biology or science, amphi just means both of something. Okay. So amphipathic has both hydrophobic and hydrophilic. That's the one in here, you know it, that if it falls in the lipid category, it has to have hydrophobic to it. It's just so happens that that phosphate group also gets that hydrophilic. So it falls into both. Triglycerides, you probably heard of these guys. The glycer part, if you hear glycer ever, you probably are thinking of like glycerol and or things like relating to glucose and sugars and things like that. And you're right, you should be thinking that. So glycerol is kind of more related to the sugar side of things. Has carbons and hydrogens and oxygens and things like that. But it's basically like a framework for the tails to hang off of. That's all it is. So we have 3 tails hanging off of a framework. That's all that this is. So 3 hydrophobic tails. Sometimes you just have carbons and hydrogens single bonded to one another. If there's a double bond, it gives a kink in the tail. And that's what we call unsaturated. Saturated means that you have all the hydrogens you could possibly fit. There's no double bonding. It's just all carbons and hydrogens. Hydrogen's taken up all the bond. Okay. If it's unsaturated, that means there's places where there could be hydrogens, but instead we're double bonding to a carbon. It's not saturated with hydrogen. So that's basically the definition of a triglyceride, and that these guys, based on whether they have a double bond or not, the double bond gives them a kink, and they're actually going to be less rigid if they have the double bonds in there. Gives them a little bit more fluidity than the ones that are just completely saturated. So the saturated fats, why they're bad, because they'll actually congeal and stick more and be more of a problem in creating issues with like, sticking in your arteries and things like that. Okay. This is kind of part of it. And your body will take energy to break down those double bonds too. So it's going to be, more useful for you that way as far as like your metabolism. We're not going to talk about the details of that. What you need to know in this right now for this chapter is simply that the triglycerides falls into lipids. So it must have hydrophobic aspects, right? Fatty acids, it's again, similar sort of concept with those long carbon chains. We talked about phospholipid. This is the actual chemical depiction of what a phospholipid looks like. You can see it has 2 tails coming off of it, branched off of that negatively charged phosphate group at the head. And you can create any sort of situation where this head will associate with the water and the tails are on the inside. Well, our lipid bilayer is what creates our membrane. But you can even have these guys. Well, you guys remember what this is called, this structure? Micelle. So whenever you have micellar water, we have charges that are going on in molecules in the water to help promote this sort of a thing because your lipids are like that. So it's actually more friendly to your membranes on your cells than just straight up water. And so it helps with like that hydrophobic aspect, it helps marry those 2 environments together and be a little bit more friendly. It doesn't strip you of all your oils and all of that. So if you guys have ever heard of that, it has stuff in it to help promote lipid happiness. Okay. We also have, other than just your phospholipids, we have other lipids that'll be inserted into your membrane to help give stability and shape. Our cells, as animal cells, we don't have a cell wall that gives our cells structure and shape. However, we have within our membrane more lipids that give structure. Now look at the structure of cholesterol here. You see all these rings and stuff like that. That structure there that looks like structure to us, looks like a nightmare as far as organic chemistry goes. But all the structure that's going on there gives structure to your membranes too. So it gives that structure and stability. Nice sturdiness. Not great if it's just floating around on its own in your bloodstream because they'll just like congeal together. Because what's your blood primarily? What's the number 1 component of your blood? Water, right? Right. And what's water but polar? And what's cholesterol that falls into a lipid? Hydrophobic, right? They don't want to mix with your blood. So they'll just stick together and congeal in your bloodstream and then catch up and even clog your entire blood vessels sometimes, right? Especially the really thin ones, like capillaries. or the ones that are very thin, supplying blood to the actual heart tissue themselves, like your coronary arteries. So those can get clogged very easily with things like cholesterol because they don't want to mix with the water in your blood, right? So anyways, moving on. So cholesterol is a type of steroid. It actually has it in the name if you're paying attention, steroid. So there's other steroids that are out there. Some of them are involved in your hormone structures. Some of them are involved in membranes of other organisms and whatever. But they all have that similar sort of shape with all those rings in it, and they're hydrophobic. Basically, like their main structures are hydrophobic. We also have waxes. I'm not going to go too much into waxes. There's only one wax that you're really going to have to know in this class, and that is mycolic acid. We're going to talk about it in chapter 4. Okay, moving on to proteins, the shapers of life. These are the predominant organic molecules in cells by far. Every single action that is governed in your cell will be somehow governed by a protein at some point. Okay, we would not exist or live or do anything without proteins. In fact, we see all the time talking about cells and how they function. Most people kind of get the gist, especially we're going to talk about it in chapter 5, like the organelles and like what each one does and everything like that. When we think about a protein being made in your body, you know we start with your DNA code and you pick the gene you want and you make a copy of the gene and then you make the protein from the gene and then it gets put wherever in the body where it needs to go or secreted from the body where it needs to go, right? It doesn't, but how does it get there? You can say all day long, well, it gets moved in little vesicles. How do the vesicles move in your cell? They don't just move on their own. It's not magic. So how are vesicles getting moved around your cell? You literally have proteins that physically carry the vesicles along the structures in your cell called your cytoskeleton, literally carrying the vesicles on their backs from place to place. That's how your vesicles move around. That's how things move around inside of your cell. They're not just magically hoping that they float to the place. Proteins are carrying. Now that is nuts because you don't see that in the drawings, right? But anytime anything needs to move someplace in your cell, a protein is carrying it. And what do you think needs to happen in order for that protein to carry something? You need to input energy. So anytime anything needs to ever happen anywhere in your cell, you're going to probably need to put in a lot more energy than you're probably going to get out. And where do you get all that energy? And that's what we're going to talk about in chapter 10, which is aerobic respiration. It's really important stuff. And we'll get to all that, but I just love the build up to it. Tell me it doesn't get cool when you start seeing the big picture of it. I just love that stuff. So anyways, proteins, they're doing everything in your body. These guys are made up of their monomer. So we talk about sugars, right? The carbohydrates, they're just sugars linked together, called saccharides, that's the fancy word. Polysaccharide, that's carbohydrate. What's the monomer for lipids? There's not really, we didn't really discuss the monomer for lipids, and I don't care that you know a monomer for lipids. Just know they're hydrophobic, please. Okay. All right, but proteins, now we're back to a monomer. They're made up of amino acids that are linked together. That's their subunit here. Just repeating amino acids. We have 20 different amino acids that are available in biology. And based on if you have amino acids that have a polar charge versus amino acids that have a non-polar versus amino acids that have a full-on charge, not just polar, and how those amino acids interact next to the one next to it versus the one that's across on the other side of the ribbon or whatever, dictates the shape and therefore the function of the protein. So the amino acid sequence is what's going to dictate all of that. Your genes encode which amino acid comes in the sequence, and that's what it is. And chapter 6 is our genetics chapter, and we'll literally talk about how that happens when we get to that. Okay, these are all of our amino acids. You don't need to memorize them. You don't need to draw them. You don't need to recognize them or anything like that. Okay, I'm not making you know this. I'm just showing you them so you get an idea of the 20 different amino acids. So these terms you probably heard of. Things like lysine or cysteine, or anybody ever heard of monosodium glutamate, MSG? Yeah, that's just glutamic acid. There's another term for that in chemistry because you haven't been paying attention. Something is an acid in chemistry that's organic. It also can be a base. And I'm never going to distinguish between the two. Same thing to me. Okay, in my class, for my purposes, unless you guys go into like actual biochem, to me these are not different than bases. Okay, so like pyruvate and pyruvic acid, same thing. Okay. But glutamate and glutamic acid, we're just talking about one sodium added onto this. The sodium that's kind of carried on, sort of like how when you take medication, something will be like blah 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 HCl. the HCl or hydrochloric acid is just there to help maintain the stability of it and make it polar. I don't know how the sodium works with that. It is. Okay. So these are things that are in your body that make up your proteins, and you have proteins that'll have all of these. Some proteins that just consist of mostly just one, or all polar ones, or all non-polar, or whatever. And how we mix and match these guys determines, again, the structure and the function. I'm not going to make you draw an amino acid or even recognize what the alpha carbon is or anything like that. But just for the sake of understanding what an amino acid is, it's essentially the following. It always starts with a carbon at the center. That's your alpha carbon. And then on your amino acid, you have an amino and you have an acid. Go figure. So you have an amino. If you guys don't remember what amino is, it's NH2. So here's your NH2. And on the other end, you have acid. So your acid in this case is a carboxylic acid. So that's always a CO2. And then in your alpha, just to make your carbon happy, you always have 4 bonds, right? So always make sure you have 4 on your carbons. It's attached to something. And that R group is what determines if it is, cysteine or alanine or valine or glutamate or whatever it is. So that's what an amino acid is. And these guys will link together. Make myself some more space here. These guys will actually link together like so. N, C, C. This guy was a double bond. That was an O. This is an H. Just be patient with me here. I've taught myself through it, even though I have a biochemistry degree. That should make you guys feel better. Even still, somebody has a biochem degree still has to talk through these things. And this is it. H. So here we have one amino acid. Well, here we have one amino acid. I'm going to erase this line. And then here we have another one. And then I'll go on to another thing. R, H. You don't need, like I said, I'm never going to make you draw this. I'm just showing you how it works, okay? So here's one amino acid, here's another amino acid. This bond here between the 2 amino acids, you guys know what that's called? Peptide bond. And again, I'm not going to make you draw it. I'm not even making you recognize it in a picture. But I am going to be talking about peptides and peptide bonds and amino and those things. So just remembering it and like having a sense of what it is. That's my whole point of drawing this out for you, okay? You don't need to draw it. You don't need to recognize it. Don't put it on a flashcard. Okay? You're welcome. All right, you guys go into biochem, then you'll have to know it. I'm sorry, but not in my class. All right, peptides. So peptide, so we talked about peptide bond. That's the bond that literally happens between 2 amino acids. That's what that is, literally. So a peptide is a molecule that's just composed of amino acids linked together by those peptide bonds. So that could be any number of amino acids. So like 20 amino acids, it's a polypeptide. Okay. So once you get long enough, then we're talking about actual proteins. 20 amino acids is probably not enough to make a functional protein. You're going to need thousands. So yeah, it's usually not going to be enough for just 20. We call it a polypeptide. They'll have their own functions in biology, but they won't be true proteins. They're just polypeptides, right? Let's talk about proteins and their structure. There's different levels of protein structure that we'll need to be aware of. And this is definitely one of those things that I test regularly. So you'll see it on Unit 1 exam. You'll definitely see it on the final. Okay. So there's 4 levels of structure to proteins. They are 1 through 4. So our primary, the first level, 1 degree means primary. So this is literally just the amino acid sequence. So whether you have alanine, phenylalanine, glutamate, whatever, they're all just linked together. Think of this as like the ribbon. We're going to talk about the ribbon folding up. Okay. So this is the ribbon. So whatever your sequence is of this amino acid 1, amino acid 5, amino acid 20, amino acid 19, and then all just lined up in order with one of them. Now they're nice fancy names. Possible 21. Okay. That's your primary structure. That's it. It's like how you spell it out. Next is our secondary structure. Well, if you have a whole bunch of polar guys next to each other versus hydrophobic guys next to each other versus alternating hydrophobic and hydrophilic, you can see how those R groups might want to be near each other, or how much literal physical space each R group takes up, whether a negative is next to a negative or negative is next to a positive, or whatever it is. Those chemical groups that are immediately next to one another will cause obvious changes at the very, very first stages in forming this protein. It's literally just the guys next to each other. So those first structures that we see formed that are clear characteristic structures, there's two biggies, the alpha helix and the beta pleated sheet. So the alpha helix happens just like you would think it would. It's a helix. It's a coil. That just happens from the amino acids that are next to each other, or the amino acids that are next to each other could make you do this. You see how we have lots of straightaways that are just folding back and forth? So this is the alpha helix up top, and this is the beta pleated sheet. Because we're creating a sheet just from going back and forth in straight lines. All right, so that's the secondary structure. So here's the picture of the primary. The secondary is down here. You can also just be random. It just depends on whatever the thing is next to you. Okay, but these are the big structures that you would see forming first: alpha, helix, and beta pleated sheet. So now we're into the tertiary. Basically, once we get the whole thing cranked out, how everything in that whole ribbon now is interacting with everything in the ribbon. Not just the guys next to you, but the whole big picture now. So those beta pleated sheets, how they're interacting with the beta pleated sheets way down the way. How these alpha helices are interacting with random parts down the way and whatever. So this is creating now, we're tying a bow. So you might have curled your ribbon first. That would be your secondary structure. The ribbon itself, that's your primary. Curling the ribbon, that's your secondary. Now we're going to tie it into a bow. That's your tertiary. Okay, that is your tertiary. That's it. So you're like, but wait, there's another structure level. You're absolutely right. There is. I'm going to tell you what the quaternary structure level is. You're ready to decorate your package and you want to put more than one bow on it. Putting those bows together, that's the quaternary. So now you have more than one ribbon, more than one polypeptide. And oftentimes, functional enzymes and things in your cell require more than one polypeptide to come together and make a functional protein. Okay, so more than one ribbon. And they'll come and work together as units to make the pretty bow that decorates your package and works. Right? That makes sense to you guys? All right. All right, so what do proteins do in our body? The two biggies, especially in our course. Of course, proteins can be structural. They can make channels through your membranes to allow things to pass through that can't handle moving through that weird bilayer that has charges and like hydrophobic on the inside. Like that's a weird situation for most molecules. And it's what keeps what's outside and what's inside. But you need to be able to move things in and out. And a lot of times proteins create channels in the membrane. So it's kind of a structural aspect. But we also have a lot of functional proteins too. This our enzymes and our antibodies are our two biggest groups that we'll be talking about. We'll talk about enzymes and different groups of enzymes and how enzymes work and everything like that in chapter 10. But enzymes are essentially proteins that help your body overcome the activation energy needed for a chemical reaction to occur. So you need to break down a molecule into smaller parts. Great. This protein is going to make that easier for you. It's going to help you with that. It's sort of like instead of trying to use your own brute force to break apart a brick wall with your bare hands, using a sledgehammer. The enzyme would be the sledgehammer. Or using an actual like excavator. The enzyme would be the excavator. You get what I mean? So these enzymes are going to be what's going to help with that stuff. So they overcome the work. They help make the work a lot easier for the body. So they're catalysts. And catalysts don't have to be organic molecules like enzymes. But in this case, enzymes are organic molecules that help overcome the energy needed for something to happen in the body. Okay, so antibodies are something else. They're essentially little Y-shaped proteins that your body makes that bind specifically with some foreign marker. So if your body can see it as foreign, it's non-self, then it can make a protein against it. In fact, your body can make 8.5 billion different possible antibodies. You only got 20,000 genes. And that is impressive work by your body, I gotta say, because usually you need a gene for every protein in your body. That's not the case with an antibody. So your body likes to play splicing games with your gene in those cells that make antibodies or B cells. And so that's how we get that variability. And thank goodness for it. And there's all sorts of ways that we sort out like the anti-self guys and get rid of ones that don't work very well or are useless. But because of course there's going to be that variability. But the ones that come out in the end, these are the guys that help give you protection that once now I've had chickenpox, I can't get it again. If I see chickenpox again, I've got antibody already being cranked out by myself so fast, I never knew I saw it. It's why you guys can go to the fair and to the mall and whatever and not get measles from people that probably have it. Because you got the vaccine, you're already making antibody. The second your body sees it, it's like, got it, I got it. You get no symptoms and you never know it was there. Your antibodies are that effective. And so, of course, we wish sometimes in some cases they'd be a little more effective, like what we have with COVID. You still get it, right? But tell me the difference now. If you get COVID now, you get cold. How many people who were otherwise healthy in the beginning of COVID before we had the vaccine, how many of those people would you see on respirators who would die? It was a huge problem. It's why we had social distancing and masking and everything like that. The only reason you're here today in this classroom with me right now is thanks to the vaccine, helping with your body prepare it so that when it sees COVID again, you have some kind of defense in place. So it's not 100% perfect. Maybe it can be improved upon, and maybe we can find a better target for those antibodies. But it works for what we needed for now. So that's the idea with antibodies. They help us see foreign things and recognize them as foreign. Proteins, and like I said, a lot of their function rests in their actual structure and not as much in their chemical makeup. Sure, the chemical makeup helps dictate structure, but if you have a polar amino acid and you replace it with a different similarly charged polar amino acid, you'll still have the same structure probably, and still have probably the same function too. So that structure is incredibly important. Its structure and its ability to then form its function as a result of that structure, that's its native state. So the native state of a protein is its functional state. If you denature a protein, it means you're taking out that normal structure of it, and now it can't function the way that it should. So a lot of the cleaning products that we have help denature protein. And since a lot of these are based on you're tying your bow, the protein will tie its own bow, by the way, just because of those chemical interactions. But as that bow is being tied by that protein that's being made, that a lot has to do with those polar molecules interacting with other polar molecules, or those hydrophobic being, pushed away from the polar guys and what have you, wanting to hide away from water and all of that. So once we have your structure of your bow made, it wants to stay that way as long as we keep it at that pH. However, you change up your pH, now you're introducing, like I said, you're interfering with those attractions. So now we can denature proteins just by changing pH. Now, you can see if it's so important that we have our structures of our protein to even just move things around in your cell. You mess that up, you kill something. And that's what a lot of cleaning agents target. Even ethanol will interfere with that. We already saw how it kind of straddles that line. It can interfere with that and cause proteins not to work properly and denature them. It's pretty cool stuff when you break down the chemistry of it. All right, our nucleic acid, this is DNA and RNA. You guys already probably know what it stands for. Ribonucleic acid, or take away one of the oxygens and you've got deoxy, ribonucleic acid. All right. These guys consist of nucleotides. That's their monomer. All right, so repeating nucleotides. And what's a nucleotide? You will have your sugar, your 5-carbon sugar. You will have that linked to a phosphate group. Just put PO4 down here. You guys already know about this guy having a negative charge. And that sugar will also be attached to a base, a nitrogenous base, which nitrogenous base literally just means a molecule that has nitrogen in it. It's got some ring action going on too. Guess what? That's all you really need to know about it. Okay, it's got nitrogens in it and carbons and hydrogen and oxygen. All right, those oxygens, again, remember how we love an oxygen having an electronegative aspect to it? This is what's going to create those hydrogen bonds between the two sides of your DNA. And it's what makes A want to bind with T and C want to bind with G. We're going to talk a lot about that in chapter 6. But A is adenine, T is thymine. And I always think of them as the ones that have like the crossbars, like they have a very structured, severe look to them, whereas the C and the G have the round. Is that's how I know the rounds go with the rounds and the structure go with the structure. I don't care that you know if they're purines or pyrimidines or anything like that. I don't care about it, okay? This slide talks about it. I'm never going to test you on it. I don't remember which one is which most of the time. Okay, all right, but we do know that you have your repeating sugar. This is D for deoxyribose, okay? Sugar, phosphate, sugar, phosphate, sugar, phosphate on that backbone. And then in the middle, we've got our bases. So the A binds to the T and the G binds to the C. And then basically, whether you have A, T, C, G, A, T, C, G, A, T, C, G, encodes essentially what amino acids you're going to make. We'll talk about how that works in chapter 6. You don't need to know those details yet. But this stuff, this is your Smithsonian Institution contents. This codes for anything that your body could ever possibly want to make, ever. And that's all in your DNA, all coded by just A's, T's, C's, and G's. All right, RNA, ribonucleic acid. This is like if your DNA is your book, your old book that has all the instructions legit for everything your cell could want to make. Your DNA, your RNA is you coming in there, writing down the instructions for how to build a chair, and then taking it home with you to build your chair. So RNA is a working copy of a gene. So you pick the gene you want, make a working copy of it, and then take it and go work with it. So that's all RNA is. It also makes up the structure of ribosomes. That'll be the machines that read that and everything like that. Like I said, that's a chapter 6 thing, but so that you get the gist of it. ATP is another one that falls into our nucleic acid category. It's our energy molecule. It's adenine, or adenosine. So it's adenine with a sugar that makes the adenosine, and then triphosphate. So it's our nitrogenous base, the same A that's in DNA and RNA. Okay, with a sugar. It happens to be ribose with 3 phosphates on it. And man, guys, so we're talking about this situation here. Here's your A. This is your A up here in the orange. This is your A. This is your sugar, your ribose. Now look at these guys. This is negative charge. This is negative charge. This is negative charge. 3 phosphates. Yeah. Now, each of them has a negative charge. If I put 3 south poles of magnets all next to each other, are they going to like that situation? They're almost going to be pushing apart, right? Negative pushing apart against negative. Those covalent bonds are forcing them to stay together, forcing them to share their electrons with one another. It hates it. It doesn't want to be like this. It wants to kick off one of those phosphates. Let's kick off both eventually, but it really gets a lot of energy released if you kick off one of those phosphates. This is the #1 energy source in your cell. Your cell will create just shitloads of ATP to drive everything that's going to go on in your cell. It is the #1 source of energy in your cell. If you run out of ATP, you die. In fact, if I were to have you guys hold your breath and just say, hold your breath, keep holding it, keep holding it. The reason that you feel like you need to take a breath in is to drive making ATP. You're like, well, I need oxygen. Yeah, what do you think the oxygen's for, buddy? Making ATP, making energy for your cell. Because you are constantly doing things in your cell. You always need to be driving that. So you need ATP so bad that within minutes you'll die. That is insane. We'll talk about just how cool and how insane that is in chapter 10. Like I said, it's just I get so antsy about it. I just love talking about it. But ATP, it's one of those things I can't stress enough the importance of this molecule in your body. Super important molecule. It is technically a nucleic acid molecule. Okay? All right, we got class questions. We do have time to go through them really quick. Lexi, you knew you were up first. Your first question is, explain what a covalent bond is in your own words. Where's Lexi? All right.
It's a bond between atoms that share electrons.
Exactly, sharing electrons. You guys hear that? Great answer. All right, what types of molecules tend to be hydrophobic? This is my question. So molecules that are hydrophobic are going to be non-polar molecules. You see, they look like lipids stuff, right? Carbon, hydrogens, non-polar. All right, our next one is for Erica. What is pH a measure of?
It's concentration.
Exactly right. So concentration of hydrogen ions, even if you had said like it's a measure of the acid, I probably wouldn't get too picky of it as long as you understand acid's a measure of hydrogen ions, right? And it's that shift based on water being 7 and what side you're on that 7. What major and essential biological role do phospholipids play in the cell? This one's Fabiola. Are you, oh, gonna show you. Like, I hear your voice. It was like disembodied for me. Yes, so my membrane structure, exactly right. So phospholipids are incredibly important in our lipid bilayers. And then our last question, the monomer subunit that makes up protein Levi. Amino acids, exactly right. So it's just repeating amino acid sequence. So you guys get how that's going to work now. You have plenty of time to prep for your question. You know it's coming up and everything. And even if you have it in the general ballpark, I'm going to give you a thumbs up probably, even if you're just engaged in a conversation. Okay. So that's we're going to head up to the lab next. And I'll see you guys up there. We don't start lab until 11:30. So like I said, be up there by 11:30. We're going to start off with a Kahoot, just like we do with the lecture. And we're going to start off with that Kahoot, and then we'll get into talking about how the course works and everything like that. It is required attendance day, okay? Yeah, and it's just upstairs, and you'll have to like go, there's construction up there. If you guys have been up to the physio lab, you know, but there's construction up there. Go on the left side of the construction to get to the micro lab.